If you have been eating less, moving more, and still watching belly fat stay exactly where it is, there is a biological reason for that, and it has nothing to do with willpower. After 60, visceral fat, the fat stored deep inside the abdominal cavity around the organs, operates through a completely different set of mechanisms than the fat that responds to simple calorie reduction. It is driven by hormonal shifts, chronic inflammation, and changes in how the body signals fat storage at the genetic level. Cutting calories addresses none of these mechanisms directly. The 9 foods we are covering today do. Each one targets a specific biological driver of visceral fat through a documented mechanism. Together, they shift the internal environment in which visceral fat either grows and persists, or cannot. Why visceral fat after 60 is biologically different. 3 changes after 60 make visceral fat significantly harder to address through standard approaches. First, hormonal decline. When estrogen and testosterone fall, the body preferentially redirects fat storage toward the abdomen. This is an ancient survival mechanism. Central fat was historically the most accessible energy reserve. Your biology is not malfunctioning. It is responding to hormonal signals the way it was designed to. But in a modern environment with consistent food access, this response creates a health liability rather than a survival advantage. Second, chronic inflammation. Visceral fat is metabolically active. It secretes inflammatory proteins called cytokines, including interleukin-6 and TNF-alpha. These cytokines increase insulin resistance, slow metabolism, and critically, make visceral fat itself harder to break down. The fat creates the very conditions that protect it from being lost. Third, sarcopenia, the progressive loss of muscle mass that accelerates after 60. Less muscle means a lower resting metabolic rate, less glucose disposal capacity, and a metabolic environment that favors fat storage over fat burning. Visceral fat actively accelerates this muscle loss, creating a compounding cycle. The 9 foods below interrupt these 3 processes: hormonal, inflammatory, and metabolic, at the specific biological points where visceral fat can be addressed most effectively. Food number 1. Avocado. Avocado is rich in oleic acid, a monounsaturated fat that activates a gene pathway called PPAR-alpha, which signals the liver and muscle cells to use fat as fuel rather than store it. A study at Penn State University found that 1 avocado per day over 12 weeks produced measurable reductions in visceral fat compared to equivalent calories from other fat sources. Avocado also provides soluble fiber that feeds gut bacteria responsible for reducing systemic inflammation, addressing the second mechanism directly. Half an avocado per day, added to eggs or a salad, covers this consistently. Food number 2. Blueberries. Blueberries contain anthocyanins, flavonoid compounds that suppress the inflammatory cytokines, specifically interleukin-6 and TNF-alpha, that visceral fat secretes to sustain itself. Human studies show that regular anthocyanin consumption reduces circulating inflammatory markers measurably over 8 to 12 weeks. Anthocyanins also improve insulin sensitivity, directly addressing the insulin resistance that drives abdominal fat storage. 1 cup per day, fresh or frozen, the anthocyanin content is equivalent in both forms. Add to yogurt or eat plain. Food number 3. Fatty fish, salmon, sardines, mackerel, and herring are rich in EPA and DHA, long-chain omega-3 fatty acids that activate a hormone called adiponectin. Adiponectin circulates through the bloodstream and signals visceral fat cells to release stored energy for burning. Low adiponectin is one of the most consistent biological markers of high visceral fat. Omega-3s raise it. Simultaneously, EPA and DHA are among the most potent natural suppressors of the inflammatory cytokines that visceral fat relies on to persist. A study in the International Journal of Obesity found significantly greater visceral fat loss in adults consuming fatty fish twice weekly compared to equivalent calorie controls without fish. 2 to 3 servings per week. Canned sardines in olive oil are equally effective and combine 2 items from this list simultaneously. Food number 4. Green tea. Green tea contains EGCG, epigallocatechin gallate, one of the most studied natural compounds in visceral fat research. EGCG activates an enzyme called AMPK, adenosine monophosphate activated protein kinase, which functions as a master metabolic switch. When AMPK is activated, cells shift from fat storage mode to fat oxidation mode, with the effect concentrated specifically in visceral adipose tissue. A randomized controlled trial in the American Journal of Clinical Nutrition found 17% greater visceral fat reduction in participants consuming EGCG-containing green tea daily over 12 weeks compared to placebo. 3 cups daily without sweetener. The sugar cancels the metabolic benefit. Food number 5. Cruciferous vegetables. Broccoli, cauliflower, kale, Brussels sprouts, and cabbage address a driver of visceral fat that is almost never discussed, the aromatization cycle. As we age, in both men and women, visceral fat tissue converts testosterone into estrogen through an enzyme called aromatase. More visceral fat means more aromatase activity, which means more estrogen, which drives further visceral fat accumulation. The cycle sustains itself. Cruciferous vegetables contain indole-3-carbinol, which converts in digestion to diindolylmethane, DIM. DIM supports healthier estrogen metabolism by directing excess estrogen toward a less potent metabolite rather than the form that drives fat storage. It disrupts the hormonal environment visceral fat depends on. 1 serving daily. The preparation method matters far less than consistency. Food number 6. Eggs provide the highest quality complete protein available from a whole food source. All 9 essential amino acids in proportions closely matching human metabolic needs. Protein has the highest thermic effect of any macronutrient, meaning more calories are burned digesting it. It also suppresses ghrelin, the hunger hormone that tends to be chronically elevated in older adults, reducing the overconsumption that sustains visceral fat. Equally important, eggs are one of the richest dietary sources of choline, a nutrient deficient in a significant proportion of adults over 60. Choline is essential for the liver's ability to package and export fat. A choline-deficient liver accumulates fat rather than clearing it, directly contributing to both fatty liver and the metabolic conditions that sustain visceral fat. 2 to 3 eggs per day. The yolk is where both the choline and the fat-soluble nutrients live. Discarding it removes the primary reason eggs are on this list. Food number 7. Garlic. Raw garlic releases allicin when crushed or chopped, a sulfur compound with documented effects on cortisol, insulin, and inflammatory signaling. Cortisol is one of the most direct hormonal drivers of abdominal fat accumulation after 60. When cortisol is chronically elevated, from poor sleep, stress, or inflammation, the body specifically directs fat storage to the visceral compartment as emergency energy reserve. Allicin has been shown to reduce circulating cortisol and improve insulin sensitivity, both of which directly affect where the body chooses to store fat. 1 to 2 cloves daily. Crush or chop, and allow to rest for 5 to 10 minutes before eating or cooking. This is the window during which allicin forms. High heat applied immediately after cutting destroys most of the active compound before it develops. Food number 8. Legumes. Lentils, chickpeas, black beans, and kidney beans have a low glycemic index. They produce a slow, modest rise in blood glucose and a proportionally small insulin response. This matters because chronically elevated insulin is one of the primary hormonal signals that instructs the body to store fat centrally. Keeping that signal consistently low, meal after meal, changes the hormonal environment in which visceral fat either grows or does not. Legumes are also among the richest sources of resistant starch, a prebiotic fiber that feeds beneficial gut bacteria. Gut microbiome diversity is independently associated with lower visceral fat levels in multiple large human studies, separate from total caloric intake. A healthier microbiome produces short-chain fatty acids that reduce the systemic inflammation sustaining visceral fat. 1/2 to 1 cup per day in soups, stews, or salads. Food number 9. Extra virgin olive oil. Extra virgin olive oil contains oleocanthal, a polyphenol that inhibits the same inflammatory enzymes targeted by ibuprofen, without the gastrointestinal risks associated with long-term anti-inflammatory drug use. This chronic, gentle anti-inflammatory effect directly undermines the inflammatory environment that visceral fat creates and depends on to persist. The PREDIMED trial, one of the largest cardiovascular nutrition studies conducted, found measurably lower visceral fat in participants consuming extra virgin olive oil daily compared to low-fat dietary controls, 2 tablespoons per day over salads or cooked vegetables. Extra virgin specifically, refined olive oil loses the oleocanthal and polyphenols that produce this effect. How these 9 foods work as a system. Each food addresses a different layer of the same problem. Avocado and olive oil shift fat metabolism at the cellular level through PPAR-alpha activation and chronic anti-inflammatory action. Blueberries and fatty fish reduce the inflammatory cytokines visceral fat secretes to sustain itself. Green tea activates AMPK, the master metabolic switch toward fat oxidation. Cruciferous vegetables disrupt the hormonal aromatization cycle that sustains visceral fat in both men and women after 60. Eggs provide choline for liver fat metabolism and protein for muscle preservation. Garlic reduces cortisol and insulin, the 2 hormones most directly responsible for abdominal fat storage. Legumes stabilize blood glucose and insulin response while feeding the gut microbiome. Used together consistently, they create a nutritional environment where the biological conditions sustaining visceral fat are progressively weakened. You do not need to add all 9 immediately. Start with 3 that fit your current eating pattern. Build from there. A practical daily structure that incorporates most of these naturally. 2 to 3 eggs with half an avocado and a serving of cruciferous vegetables in the morning. A cup of green tea mid-morning. Fatty fish or legumes at lunch or dinner with extra virgin olive oil dressing. A cup of blueberries as a snack. 1 to 2 cloves of crushed garlic are added to whatever you are cooking in the evening. This is not a restrictive protocol, it is an additive one. You are not removing foods you enjoy. You are consistently adding the ones that shift the internal environment visceral fat depends on. 30 days of consistency is enough to notice measurable changes in energy, in how clothing fits, and in the blood markers associated with visceral fat. The deeper changes in inflammatory markers and hormonal balance build over 3 to 6 months of sustained practice. If this gave you a clearer picture of why visceral fat behaves differently after 60 and what specifically moves the needle on it, please share it with someone who needs this information. Subscribe for more evidence-based health content designed specifically for adults over 60. We will see you in the next one.